there I am on the monitor. And I gotta tell you, I look like somebody. I just can't put my finger on it. But anyway, so Will Nye here, Will Nye the other guy, and I've had way too much caffeine today. And today we're going to be looking at the subject of abiogenesis. What is abiogenesis? Abiogenesis is the study of non-living material going to being turned into the first living cell here on planet Earth. So there's an entire study about that, and we're going to see what kind of progress has been made in that area. So, first of all, when we're talking about the building blocks of life, the first thing we want to look at is we want to look at something called Zdica-onema. Zdica-onema 02. Wait, hold on for a second. We want to look at the 20 amino acids that are coded by DNA. Now, there's all kinds of amino acids out there beyond the 20, but as far as what occurs in nature, what's coded for by the genetic material within a living cell would be 20 amino acids. And they come in two configurations. There's a left-handed and a right-handed. We'll get to that in just a second. And you got to have those. In a living cell, you got to have protein, you got to have some genetic material, and you got to have a way to make the proteins. So, Stanley Miller, back in 1952, along with some other people, put together an experiment where they take the constituents of the theoretical early Earth atmosphere, which would be water, methane, ammonia, and hydrogen, and they have a trap for the whatever they make in their little experiment, and so they zap the vapors and they end up with amino acids. Now, at the time, back in 1952, Miller said, hey, look, under early Earth plausible conditions, I've come up with 11 of the 20 amino acids, and everybody thought that was really good. So it's a little bit of a problem because, you need all 20, but he comes up with 11. He also comes up with some that don't exist in nature, and they're left-handed and right-handed. We'll get to that. You need to have, well, actually, let's just talk about it right now. You have to have all left-handed, okay? So here's our early Earth, and we got our constituents, and we come up with left-handed and right-handed. We come up with some proteins that some amino acids that we don't need, and we come up with at least 11 that we do need. Well, come to find out, years later, they took a look at some sealed vials of what was actually produced back in 1957, and they actually were all 20. But the other ones beyond the 11, which would be 9, basically were in trace amounts. In other words, almost non-existent, but they were actually there, which is a good thing. Anyway, so proteins within the cell are used for enzymes, are used for structural material and other things like that, and they have to be folded into different shapes to perform a particular function. Like, for instance, if it's going to be an enzyme, it's going to need possibly to bind with another molecule, and so it has to fold into a particular shape with a particular binding site exposed, and it goes and has to link up with a different molecule. If in your link of 11 amino acids you have a right-handed type in with all of your left-handed types, it won't fold up and it becomes useless. So the Miller experiment doesn't exactly tell us how it is that we came up with all left-handeds, when that's what you need, but at least we came up with some amino acids. So it's a very interesting experiment. Doesn't quite tell us how it's done, but it's a possibility. So and at this point in time, scientists are actually going beyond that and looking for other ways it could have been done. Because really we've got to come up with all 20, and we have to have some way of coming up with only left-handeds. So there's different experiments that basically seem to insinuate that possibly you could have conditions where you might have a slight leaning toward left-handeds, but you still have some right-handeds in there. So that's not quite good enough, but we're actually getting there. So, moving on. The winner is, of course, the left-handed. So, let's see. We also have the same problem when you have DNA. DNA, is all right-handed, and if you have a left-handed link in your DNA, you don't end up with your spiral, your double helix, and that doesn't work right. So it's going to be a problem. The first living cell. So let's think about what the first living cell would have been like. What kind are we going to come up with? So we're probably not going to come up with a really complex one that has all kinds of organelles. We're going to come up with probably the simplest one we can think of, which would probably be a bacteria. So bacteria falls in the category of prokaryote. Okay. Now there's another type that's more complicated than that, but we're going to deal with prokaryotes, which would be a very simple organism like a bacteria. And it should at least have a cell membrane. It's going to have some, like a jelly-filled donut, so there's going to be the inside where it's got cytoplasm. We're going to need a ribosome to make the proteins. And you're going to have a nucleoid, which is just a place where you have a single strand of DNA, or you might have only a single strand of RNA, which makes the processes within the first cell a lot simpler. Next, ribosomes. It's a complex molecular machine that's found inside of living cells, and ribosomes are used to link the amino acids together to form a specific protein, and this is done in conjunction with messenger RNA, another molecule that we're going to need within the cell. Ribosome. Write it down. There's going to be a quiz. The earliest known living cell would probably have to have at least 200 proteins. That's a theory that's being kicked around out there. That's really a lot of proteins. Within human physiology, the simplest protein has 44 links. The most complex one has 34,000 plus links. So let's see. Wikipedia. I had to do some research here because that actually ended up pretty good because it really boiled it down to what we're looking for, because we have to come up with a theoretical first living cell, so we need to kind of taper it down to something that's really manageable and simple. So Wikipedia, let's see, says that the average size bacteria contains about 2 million proteins. That's going to be too many. Okay, but the simplest one has only 50,000. It's going to be too many. Okay. And of course you're going to have DNA that's going to code for all of those proteins. That's a big job. So, let's get down to it. There's 20 different amino acids. The proteins do a specific, I can't say that. Proteins do a specific function within the cell. So, when you're linking up amino acids to make a useful protein, you look at each and every individual link. And so if you're trying to come up with a particular protein, like an enzyme, you look at link number 1 and say, well, it's got to be phenylalanine, or maybe it needs to be lysine, or maybe it needs to be proline, but it needs to be whatever it needs to be. So there's 20 of those, so we have to come up with the correct one. So you have 1 chance in 20 of your first link being correct. Then you're going to have a second link. You have 1 chance in 20 that your second link is going to be correct. So what are the chances that both of them are going to be correct? The first one and the second one. That would be 1 chance in 400. Then when you go to link number 3, you end up with 1 chance in 8,000. You go to link number 4, you end up with 1 chance in 160,000. Link number 5, you end up with 1 chance in 3.2 million. When you get to link number 6, you end up with 1 chance in 64 million. When you go to link number 7, you end up with 1 chance in 1.38 billion. And when you get to link number 8, you end up with 1 chance in 25.6 billion. And when you get to link number 9, you end up with 1 chance in 5.12 billion. And when you end up with link number 10, you end up with 1 chance in 10 billion. Now, you also probably want to factor in left-handed and right-handed, because in nature, as we were talking about in the Miller experiment, you end up with left-handeds and right-handeds. You have to have either all of one or all of the other. And every now and then you might get one in there. Maybe you're going to get away with it. But for ones that have a specific function where it needs to fold, it needs to fold in a particular way to do a specific function, you can't have any right-handeds in there. So, to make the math easy, having 20 amino acids that can be left-handed or right-handed, that's like having 40 different amino acids, because you get 2 prolines, a left hand and a right hand. You get 2 phenylalanines, you get a left hand and a right hand. Okay, lysine would be the exception, but we're going to make the math simple, and we're going to call it 40. So, what's the chance that your first link, your first amino acid is going to be the correct one for what you're trying to get? And we'll get to that, what you're trying to get, in just a second, because that might be too stringent of a way of looking at it. But anyway, so at link number 1, it's 1 chance in 40. You can see it okay, right? We're going to get to that. It's a big number. Okay, and then for link number 2, it's going to be, it's for link number 2, it's 40 times 40, which is 1 chance in 1,600. Link number 3, 1 chance in 64,000. Link number 4, 1 chance in 2.6 million. Link number 5, 1 chance in 102.4 million. Link number 6, 1 chance in 4 billion. Link number 7, 1 chance in 164 billion. Link number 8, 1 chance in 6.5 trillion. Link number 9, 1 chance in 256 trillion. Link number 10, 1 chance in 10 quadrillion. Okay. 1 chance in a number which is a 10 followed by 15 zeros. Okay, so I went to this website. I believe it's called The Physics of the Universe by Numbers or something like that. I'll get to that in a second. The distance from here to Proxima Centauri in meters. The number that I just was talking about, a really big number, 10 followed by 15 zeros, would cover 1/4 that distance. Or another way of looking at it is if you were to take that number and say those are meters, what's the distance of a 10 with 15 zeros after that? And we're talking meters here. What's the distance? That would be about 1 light year. Okay, so that is to try to get a protein that's 10 links long in the correct order and all left-handed. Yeah, simple stuff. It's easy. Don't even worry about it. Okay. Oh, that's that's that big number. Okay. So now we have to come up with a model. This is a mathematical model. Okay, it doesn't really exist. It just exists in our minds. Okay, the first theoretical cell. So, okay, let's take something that exists in nature. Let's take, a protein. 200 million. No, we're not going to go with 200 million. How about a really simple, bacteria? 50,000 proteins. No, we're not going to go with that. So let's go with 10,000. Okay? No. Let's go with 1,000. Dude, you're harshing my buzz. It's going to be way too hard. It's going to be hard. So the math is going to be too hard. Why don't we go with 200? No. Why don't we go with 100? Okay, make it easy. Okay, then let's see. How long is our protein chain going to be? So we're going to have 100 proteins. Okay, not 2 million, not 50,000, not 10,000, not 1,000, not 200. We're going to have 100 proteins to make up this first theoretical cell. It's going to be a really simple cell. It's just going to sit around and eat, like me, on a Saturday night, watching TV. Oh, it'll have to reproduce too, so we'll get to that. But anyway, so how long should those amino acid chains be? 40,000 links long? Okay, because, in human physiology, the simplest one is 44,000. You got hemoglobin that's got thousands and thousands and thousands. Okay, but this is just a simple cell, no hemoglobin, really simple stuff. So what are we gonna do? We're gonna do 44 links? 44 links, not 44,000, not 4,400, not 440. 44? You're going to do 44? 44? Takers? Anybody? 44. Do I see a 44? 44. No takers for 44. How about 30? 30. Do I see 30? I don't know 30. It's going to be too hard. You can't. Is it 20? No? How about 10? Because we already were dealing with 10. Why don't we deal with 10? Okay? Will that work for you? Okay, let's deal with 10. Okay, so we're going to have 100 proteins, and each one of them are 10 links long. Now, this will make the math really, really simple. That's 1,000 links, right? Okay, that's 10 links long and 100 proteins. That's going to be 1,000. Okay, so let's see now. Let's do some math. Well, it's actually pretty simple because, see, the thing is that you're going to end up with a number that's going to have, wait, we have to deal with one other thing. I almost forgot. Because there's going to be people out there, and I've actually talked to people, and they've said, well, when you come up with the first living cell, there's probably a lot of ways you can do, like an enzyme that does a particular thing. There's probably a lot of ways you can do a structural molecule that, does something, And so you're saying you got to have the specific one. Okay. Well, okay, so let's say for, a particular kind of enzyme within the cell, or like this protein or whatever that's going to actually help with the process of transcription from the genetic material to try to make protein within the cell, because it has to make proteins and has to grow. So let's say that for each one of the ones that you're going to need, there's 1,000,000 ways to do it. That'd be a 1 with 6 zeros. So that's sort of like going back to our original math with 10 amino acid links. And instead of dealing with 10, we should probably deal with 4. So our first theoretical cell should have 100 proteins, and each one of them will be 4 links long. It's going to be too hard, because that's going to be 40 times 40. So let's just forget the left-handed thing. We'll have to deal with that later. I can't really deal with that right now. I'm just dealing with so much right now that I can't deal with that. So it'll be, 20 times 20 times 20. So let's see, where were we? Okay. So let's see. So we got 20 times 20 is 400, and we got times 20 is 8,000, times 20 is 1,000, excuse me, 160,000. Did I that right? Okay, 20 times 20 is 400. 20 times 20 is 20 is 4K. 20 times 20 is 1,000, excuse me, 160,000. So that'd be 160,000, and then it'd be 160,000. So that's 160, and then 000. Okay. And then, of course, there's 100 of them. So basically what you end up with is because we're trying to get 100 of these, 160,000, and then, and then times 100. Okay, so in the end, we're actually looking at, except for the fact that it's 4 lengths long. So it's basically, it's going to be a number that's going to have, 400 zeros after it. Yeah. So anyway, so it's going to be a really big number. Okay, so how big is that? Okay, so after the video you can check my math, but it's a really big number, and it's going to have 400 zeros because it's 4 lengths long, and you do the math, you end up with 160,000, and you're going to have to do that 100 times, and so you're going to end up with a number that has 400 zeros after it. So let's see. Went to physics of the universe, and so let's see. There's this number that has 80 zeros after it, not 400. It's got 80 zeros after it. Okay, and that number represents, it's an estimated number of the fundamental particles in the known universe. Some people say that it's a little bit bigger than that, so it should be a number with 85 zeros after it, a much bigger number. We're dealing with one chance in a number with 400 zeros after it, okay? Because we're dealing with 4 links, and we're actually dealing with 4 links, and there's 100 proteins, so that's 400. And every time you add a link, you add a 0. That's how the math works. You can check me. Anyway, so the thing is, we have a number that has 400 zeros after it. I can't even wrap my brain around it. I mean, I got notes, and I can't wrap my brain around it. Okay, the estimated number of all the fundamental particles in the known universe. That means you got galaxies out here and galaxies out there, and way out there you got galaxies, and theoretically it's really, really, really big. And, each galaxy has a star that's huge. It's gigantic. Our sun is really kind of small. So you have like a really big, you got big suns, you probably have a bunch of planets floating around, and they have molecules. Each molecule it's got electrons and protons and neutrons, and there's probably particles that are smaller, and it's the estimated number of particles, not molecules, not atoms, particles in the known universe. 85 zeros long. That number is really big. A number with 85 zeros after it. This is a number with 400 zeros after it. Now, if you're actually trying to figure out what the chance of coming up with an actual bacteria would be, it's just so big that you would probably you'd have like page after page after page after page of zeros. It's just huge, okay? And after everything we've been through, this is legitimate. This is reality. This is just the way it is. Okay, then you're going to have, in the first living cell, you're going to have to be able to make more of these theoretical proteins. So there's some scientists that think, well, it probably wasn't DNA in the first living cell. It was probably RNA. The reason being is because you need RNA to do the transfer. So if you have RNA everywhere, you got RNA for the genetic material, and you have RNA to do the transfer, and you've got the ribosomes that are going to make the proteins, it makes things a lot simpler. I can go for that. But the thing is that the genetic code is very specific. In other words, you've got the genetic material, it codes, it does other functions, but it codes for making specific proteins. And so you're going to have a genetic code, and it's going to have to code for the cell that you're making. So you can make it bigger, and it can split, and you can have more. So all the math that we went through, you got to go through the whole thing to come up with the genetic material. In other words, there's all kind of ways to get all the links wrong, the nucleotides. So you can have all kinds of wrong nucleotides, but you can only have one right nucleotide. Once you get rid of like the million types, it would be correct, and you still have to code for 10 links long times 100, 400 links of correct amino acids to make up the entire protein situation. So it's everything we talked about already. Times 2. Okay? There we go. I don't know if I showed you that, but there it is. Reproduction. This is a family show, so we're not really going to get into it too much. But the thing is that you got to have the genetic material right there at the same time. So for the first cell, it's just going to sit there. Plus the fact that it's dead, it has to come to life, and then it has to start metabolizing, and it has to like grow, and it has to split. So you can have to have both. So that's going to be really hard. And right now, there's not really a Miller experiment that corresponds to proteins when you're talking about genetic material, and there seems to be some problems because they're talking about, well, we might be able to have this happen in outer space because it doesn't look like it's going to happen here, but somehow it happened. Okay, there you go. So first cell, no DNA, we just got RNA, and then it's got to be at the same time, and bam, first cell comes to life, and then bam, successive generations with the RNA to produce more of the same kind of cell by growing and splitting. It's easy, no problem. Okay, so we've got to put all the pieces together. So to have our cell, even though it's such a tiny little bunch of material, 100 proteins, maybe not even enough to get a ribosome or two and some cell wall. We might need some lipids also. That's another story. You got to put all the pieces together. So, and for that, I just have a picture because I don't know how you do that. Because the thing is, you don't get proteins outside of a cell. Now, there's some theories being kicked around. One of them is about how they might self-assemble on some clay, but under ideal conditions in a lab. Last I heard, it's not happening yet. But anyway, so they're working on it because that's what they do. You have to go from dead to alive, and then you have to grow. And then, of course, in the early Earth scenario, there's no free oxygen, so ozone is just like zapping everything. You're harshing my buzz. Bites. Then after that, of course, you're going to have to, your first living cell with only 100 proteins that are each one only 4 links long, they're going to have to mutate. Now, the mutations happen in the genetic material. So the genetic material is going to have to get bigger because it's going to have to code for bigger proteins. We know that. And more of them, more varieties. We know that. And so when you're doing your mutations, it's not just that the links are changing, but we're getting more links. So in other words, doing some kind of a copy error kind of a thing, you're going to have some more like pieces kind of stuck in there someplace. And that very next generation needs to be viable. So you're going to have a bunch of copies that just never work because we've already seen from the math that there's a lot of ways to do it wrong. There's not very many ways to do it right. And so that's going to have to happen. But the good thing is, okay, so you have a ribosome and it assembles the proteins. It does it at something like 20 links a second. That's almost as fast as frames of a film. Not video, but film. 24 frames a second. This is like 20 or something like that. Anyway, it's about the same. So it's like nail gun. Okay, so with only 4 links long, or if you want to do one where it's got only 10 links long, or whatever it is, it's like, it's just spitting those things out. So you should be coming up with enough material to make another protein, or a theoretical one, 3 times a minute. And you're going to need that because you're going to need lots and lots of generations of getting it wrong. Copy errors, that's the one that goes in the trash. That one's dead. That one doesn't work. That one's a step backward. Yada, 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 yada. Oh, here's one. Here's a good one. Okay, now we have yet another either 4 link long or 10 link long or 20 link long protein that does something useful that is compatible with what we already have already. It's easy. It's not going to be a problem. No problem. So, and that's where we end off at, because after that, then you end up with something called natural selection and Darwinianism, and that's an entirely different subject. It doesn't have anything to do with this. You don't even think about it. Just think about this. And there you have it. See you next time.